So this video is going to be my introduction to carbohydrates. However, if you also watched my 1.2 and 1.3, I use carbohydrates to explain dehydration synthesis and hydrolysis. So it does have a little bit of an overlap with that previous video. But anyway, let's go ahead and talk about carbohydrates. So the monomer and polymers of carbohydrates is a macro molecule, a large organic molecule, is going to be glucose. Now, glucose is a 6-carbon molecule, and it does result in a hexagon shape. And so when we look at the molecular formula of glucose, it is C6H12O6. Now, glucose is one single sugar, and the fancy scientific word for one sugar is monosaccharide. Mono means one, and saccharide means sugar. Now, glucose isn't the only monosaccharide in plants or in carbohydrates. While glucose is the main primary one that we talk about, there's also going to be fructose, which is found in fruit like honey or grapes. And then we have galactose, which is found in breast milk. Like you can kind of see the word lactose there. Anyway, so when we look at what do these all have in common? Well, they're all one single sugar. They're all made of C, H, and O. And their molecular formula is C6H12O6. Now is that a coincidence? I don't think so. When we talked in my previous video about how organisms are going to exchange matter with the environment, we learned that plants will take in carbon dioxide and water, the C, H, and O, and during the Calvin cycle of photosynthesis will produce a simple sugar called G3P. Now G3P is C3H6O3, and when you add two of those together, that's where the C6H12O6 comes from. So while we have different kinds of monosaccharides in carbohydrates, glucose is going to be the primary one that we talk about. Glucose is going to be the main monomer of carbohydrates. So let's go ahead and look at, well, how can we bond two monomers together, two glucose molecules together to begin to build a polymer? So if we have two glucose monomers joined together, two monosaccharides joined, a good name for that would be a disaccharide, with di prefix meaning two. So how do we bond them, though? That's the question. So we take two monosaccharides. In this case, I have two glucose monomers, two glucose monosaccharides. And in order to covalently bond them together, I'm going to remove a hydrogen from one and an OH from another. Now that's going to form water. So in order to build or synthesize a covalent bond between two monosaccharides, we remove a water in the process. We call this dehydration synthesis. So in order to begin to build a polymer of carbohydrates, we'll remove waters and build covalent bonds between the monomers, between the monosaccharides. Now, enzymes do this. It's not a random process that occurs. Now, in order to split apart a disaccharide, this is where hydrolysis comes into play, where we would add a water to split it. Now, let's go ahead, though, and so a disaccharide is simply two monosaccharides joined together. A really common example of a disaccharide would be sucrose, which is your table sugar. If you add sugar to your coffee, that is sucrose. Anyway, what do you think it would be called if we had a chain of monosaccharides? Right? The perfect name would be polysaccharide, many sugars. So was this polymer built, I want you to check yourself, built by dehydration synthesis or hydrolysis? Okay. So it was actually built by dehydration synthesis. And then was water required or produced? It was produced. And then how many waters? Yeah, 7. So each of those covalent bonds, a water was removed to join together the monosaccharides. So now, though, let's go ahead and think a little bit more deeply about how organisms grow, right? So if we talk about a tree, for example, we think like, how does it grow? Hopefully you're thinking to yourself, well, it's going to need to build more cells, which then means it needs more matter. And so when we think about what matter they have available to them, what macromolecules they have to build themselves and to grow, is plants are going to have carbon dioxide, water, nitrogen, and phosphorus, and they're going to use those atoms in the environment to build their carbohydrates, lipids, proteins, and nucleic acids. Now, when we talk about plants, though, a lot of the structure and components of plants are going to be carbohydrates. And so we're going to use plants as our example to begin carbohydrates, and then we'll tie it into how humans use carbs. So if we go back to glucose, glucose is a major fuel source for cells, for both plants as well as animals, bacteria. All life on Earth is going to use glucose as their primary first choice use of fuel or energy for the cell. Now, you may know from a previous biology class that the mitochondria is the powerhouse of the cell. It's going to make energy for the cell. But really what that means is the metabolism or breakdown of glucose will kind of start in the cytoplasm but be finished in the mitochondria. So we take glucose, our cells are going to take glucose, and it's going to break it apart in order to release energy in the form of ATP. Now, as we break apart glucose through a bunch of chemical reactions in the cytoplasm and the mitochondria, we break it apart into carbon, hydrogen, and oxygen in the form of water and carbon dioxide. And so we actually will return glucose back into water and CO2 in cellular respiration. So here, if glucose, though, is the primary source of energy for cells, how could a plant or animal store that glucose for later use? So hopefully you were thinking, oh, we can just join a bunch of them together into a polysaccharide. Okay. And so if we were to build a polysaccharide, we would produce water in the process of dehydration synthesis. So we're going to start with plants and their polysaccharide that they use to store energy. So plants, once they synthesize glucose, they are able to join a bunch of glucose monomers together to form this polymer, a polysaccharide, called starch. So starch is the energy storage polysaccharide in plants. And so when you eat potatoes, rice, pasta, like wheat, this is going to be the starch that the plant made to store energy. And this is why when you eat these carbohydrates, you have now extra glucose within your body that you can use for energy, right? And so when we eat starch, what do you think is going to happen to it? So hopefully you're picturing when we eat starch, that's a very long polymer. It's a very large molecule. What we will actually do, we have enzymes in our saliva that when we begin to chew like French fries or pasta or rice or bread, what's going to happen is we're going to take that polysaccharide of starch and we will actually hydrolyze it into individual glucose monomers. And now that is energy for us. So now I'm going to transition into animals and what happens when we eat starch or when we eat glucose or fructose. So let's look at how polysaccharides are formed in animals. Now I want to point out that in animals, we have blood sugar flowing through our circulatory system, right? So we have sugar, about a teaspoon's worth, flowing through our system, and that's going to be quick, easy energy for our cells. So we try to maintain homeostasis and try to keep like even blood sugar levels. So when we eat something sugary, so I know I was talking about starch, but if you have like something like a soda has 42 grams of sugar, 39 grams of sugar. Now the sugar in soda could be high fructose corn syrup. And when we look at that, it's going to be like a disaccharide. It's made of glucose and fructose. And so when we eat that sugar, what's going to happen is we will hydrolyze it into glucose and fructose. And then our liver will actually turn fructose into glucose. So when it is in our bloodstream, it'll all be in the form of glucose. So let's say I have a large soda. And so what will happen is I'm going to have a huge increase in blood sugar. Now, am I still in homeostasis? No, this is not a good state to be in. And we can talk about that in Unit 2 and Unit 4 and as the year goes on. But in an effort to return to homeostasis, a few things are going to happen. First is insulin is going to be released from our pancreas and it will communicate with our cells that, our blood sugar levels are way too high and we need to take some out. So what will happen, one of our first locations we will store excess sugar is going to be in our muscle cells as well as our liver. So we will begin to remove glucose from our blood. Now we will talk about how this happens in Unit 2 with cells. So as glucose from our bloodstream is removed and then stored in our muscle cells and in our liver, we save it as a polysaccharide called glycogen. So this right here is glycogen. Oftentimes people will get it confused with the hormone glucagon. So this is glycogen. It is a energy storage polysaccharide in animals. And while I only have 5 or so glucose monomers linked together, it actually has a branched structure within our muscle cells and within our liver. And so, as we, oops, I'm so sorry, let me delete that. So, as we talk about this, if our blood sugar was high, and then our muscle cells are at their like full capacity for glycogen, and then our liver cells are at their full capacity for glycogen, what will we do if our blood sugar is still elevated, right? If our blood sugar is still high? Well, as luck would have it, that glucose monomer is made of C, H, and O. And what we can actually do is we can actually rearrange these atoms into triglycerides, which are long-term energy storage molecules, and we can save that potential energy in our fat cells. And so organisms are able to convert carbohydrates into lipids and lipids back into carbohydrates by rearranging atoms because they're made of carbon, hydrogen, and oxygen. And when we think about food, food is a potential energy for us. And so it doesn't make sense to take it in, whether it's carbs or lipids, and then just waste it, right? This will help us in times of famine or not being able to find food. Oops, I'm sorry, ignore that. And so now what will happen though to this glycogen during exercise or just movement or carrying out daily functions? So if I go for a run, my body needs energy to power those muscles. So I will actually hydrolyze the glycogen within my cells, my muscle cells, and then use those individual glucose monomers to produce ATP to have me be able to continue running. Now, after exercise, sometimes you feel really hungry, and part of that reason is it's your brain signaling to you to replace those glucose that you used up during that exercise. So to kind of summarize so far, glucose is the monomer of carbohydrates, and the two main energy storage polysaccharides are starch and glycogen. Now, starch is the energy storage polysaccharide in plants, and plants can hydrolyze it just like we can, and they'll use their glucose in their own mitochondria to make ATP. And then glycogen is the polysaccharide that's found or stored in animals. So starch and glycogen are both polysaccharides, and they both are used for energy storage, but just in separate types of organisms. Now, there is one more type of polysaccharide to talk about, and that's going to be a cellulose that is used to build the rigid cell walls of plants. Now, cell walls are for structure and support. When you think about a plant, a tree, for example, standing up hundreds of feet without a skeleton, right? So this is going to come from its rigid cell walls. And so the structure is going to be different compared to starch and glycogen. Here you can see how the cellulose is in like chains, or well, they're all chains, but like straight lines, and that's going to be because it serves a different purpose. So now if we look at starch versus cellulose, and you kind of look at the difference between the two, and what they have in common, we see that both starch and cellulose are both made of glucose monomers. However, we notice that the bonds are different between the glucose monomers in cellulose. Now, why this matters, or the significance of this, is if I were to eat starch, I love French fries. And so if I were to have some French fries, which is potatoes, which is starch, when I begin to chew them, my enzymes will actually hydrolyze that starch into individual glucose monomers, and then I'm going to have some energy that I can use for exercise or life, whatever. However, if I have bell peppers or a salad or some broccoli that has cell walls, I am unable to hydrolyze these bonds between the glucose monomers within cellulose. So cellulose is not a good source of energy. So if I were to eat starch and cellulose in equal amounts, the starch is going to provide me with more energy because I'm able to hydrolyze it into individual glucose monomers that is usable. Cellulose, on the other hand, I cannot break apart, I cannot hydrolyze, and therefore it is not a good energy source. I would never eat a big salad before a 5-mile race. or a 5K, for example, right? Like that's not usable energy. I'd rather eat some hash browns or some rice before I have a race where I'm going to be using lots of glucose. Now, when we think about the difference, though, between starch and cellulose, sure, they're both made of glucose monomers, but the monomers or the type of glucose is different. Where starch is made of alpha glucose, cellulose is made of beta glucose, and that results in different like structures of their covalent bonds. And so we do have enzymes to hydrolyze starch, whereas we don't. Life isn't really able to hydrolyze cellulose. And so when we look at like horses or cows or other herbivores, those mammals do not have the enzymes to break cellulose. Instead, they live in a symbiotic, mutualistic relationship with bacteria inside of their gut, and the bacteria are actually going to hydrolyze that cellulose for the herbivore. Termites I don't know if termites have the enzymes themselves or if they also have bacteria, but termites are one of the few animals on Earth that is actually able to break down that cellulose. All right. So that is my summary on carbohydrates, and great job.